Okay, so today we're going to move on with our cell cycle. But before that, I'll show you guys how things looked with the exam. I don't know how I feel because the multiple choice had actually an average of about an 80, somewhere between an 80 and an 85. But once I graded the short answers, this happened. And I think really all it is just there's a few people who did awful, and that brought the average down. So overall, well, the median was a 78, so that's not too bad. So I'd say keep it up, except for those of you who are down at the tail. Don't keep that up. Let's fix that for next time. Some people who've been doing really badly did really well in this exam, so kudos to those of you. I don't know if I see some of you. Oh no, those people aren't even here. Well, kudos to the people who aren't here. You'll hear me on the recording. So after class, you can grab your exams. Okay, so far, what have we done? We went through all the phases of the cell cycle and talked about checkpoints and talked about the concept of CDKs controlling the cell cycle and how they're regulated by the availability of cyclins. If the cyclins are there, CDKs are active, and how cyclin CDK activity is regulated, right? We talked about phosphorylation, activating and inactivating. We talked about CKIs, these inhibitors that bind to cyclin CDKs and prevent them from functioning. And we talked about degradation of cyclins, right? So that's mostly where we were. And then we started to talk about regulation of S phase. So I gave you all the information about the main players, right? Those are mostly cyclins, CDKs, activating, inactivating kinases, CKIs, and ubiquitin ligases. Those would be your main regulators of the cell cycle. I always feel like I'm blocking you guys. So now, who cares what all those players are if we don't know what they're doing? So you should always be thinking about this as in, well, what's being phosphorylated? How is that affecting activity? What's being degraded? How's that affecting what phase of the cell cycle we're in, or the activity of something? What's being inhibited? We started talking about regulation of S phase, and specifically about regulation of licensing. That's that pre-RC formation and its disassembly, and regulation of initiation of DNA replication. So when you start to think about this, think about the roles of APC and SCDK in this process. And think about why with all this information you have, why does S phase occur only once per cell cycle? So just to go over where we left off. So this is what's happening, right? So you have your pre-RC formation. That's actually happening during G1. And then when your SCDK gets activated, then you end up with degradation and inhibition of some components of that pre-RC. So you get the disassembly, and then you get phosphorylation of your origin recognition complex by your SCDK, and then initiation and completion of your DNA replication. So things to think about is what's active and when are they, what is active during what phase of the cell cycle, and how is that affecting progression of S phase. So APC, right? Remember APC is ubiquitin ligase that degrades a whole bunch of things. So it is active in mitosis through G1 because it's turned on by MCDK, and since it's active, it is degrading geminin, and geminin is an inhibitor of CDT1. So that is allowing pre-RC formation. Where's my laser? There it is. Pre-RC formation, because CDC6 and CDT1 need to be available to assemble at the origin of recognition, and then that's loading on the helicase and allowing for this pre-RC formation. So as long as APC is active then you're going to have degradation of geminin, which allows this to happen. SCDK will turn off APC, and this allows for accumulation of geminine, which then will play a role in inhibiting the pre-RC and disassembling pre-RC, and that needs to happen in order for S phase to initiate. So things to think about is when is APC active, and what is it degrading, and how does that affect initiation of S phase. And then the other thing to kind of think about in this whole process is this interplay between the different ubiquitin ligases. Now, you don't have to think about it, but I find it helpful to think, well, if something's being turned off, how is it being turned off? So if you think about it, you've got your SCF and you've got your APC, and your SCF is actually going to degrade. In M phase, it's going to degrade an inhibitor of your APC, and so that's what allows it to be activated. And in late M phase, you're going to have degradation of the activator of APC, which will then just get taken over by another APC. But eventually, you have degradation by SCF of your activator of APC, which turns things off prior to S phase. Right? So it makes sense if you degrade your activator of APC, APC is not going to be active. And then you'll have, at some point, you'll also have turning off of SCF by APC, and that allows CKIs to accumulate because SCFs degrade CKIs. And then at some point, you'll also have degradation of the phosphatase that phosphorylates, that removes the phosphate group from CDH1. So sometimes, I don't know, I love protein degradation and how it regulates things. So sometimes it's helpful to think of that balance between different things being degraded, but that also might be too much for where you guys are at. So I will not ask a question like, explain the complex interplay between APC and SCF in cell cycle, because that I think is, you have to integrate way too many things, maybe for an undergraduate level class. But some people remember things more easily when they have more detail. I don't know, sometimes I do, because I always have more questions. Okay, so that's where we were stopping. And then we had these cell cycle practice problems. And we had gone through the first two, right? So how do cyclins regulate the cell cycle? Someone answered that beautifully. How do cyclins regulate the cell cycle? What do they do? They activate CDKs. So if you want to turn off your CDK, what will you do to your cyclin? Degrade it. Thank you. So you suspect that a defect in protein X has a negative effect on cell cycle progression. How would you use the tools explained in class to evaluate whether there's a defect in cell cycle and where that defect might be? And we talked a little bit about that. And to do this kind of question, you really need to understand the methods we talked about early on in this section. I think that was lectures one or two, where we talked about PI staining. And someone over here, did you say it right? I always remember the location where the answer came from. It's really weird. So PI staining and BRDU, and how you'll use flow cytometry to determine what point of the cell cycle the defect might be. And there was even, someone brought up temperature sensitive mutants. Was that you? No, somewhere over here. Temperature sensitive mutants. And how if you have a mutation, here the mutation's already there, but let's say you have a temperature sensitive mutant, you can just raise the temperature, stain with PI, do flow cytometry, and based on your DNA content in your cells when the mutation is being expressed, you'll be able to determine where your defect is, because you should have arrest of your cell cycle. Any questions about that question? Okay, we went over it the other day. Okay, so what would be the effect of a constitutively active APC? So this is an APC where CDC20, right, or CDH1, those are the two activators of APC, where for whatever reason they're not being degraded or inhibited, so that it's always interacting. What would happen if that, what would be the effect on just cell cycle or the organism? Yep. DNA would not replicate. Let's see. Right, because you would always be degrading geminin, but you would always be degrading geminin, so it would not be around to inhibit. So that would not allow for pre-RC disassembly. Yep. So DNA S phase initiation wouldn't occur. Right. Exactly. What else? We talked about. So what are some other substrates of APC that we spoke about briefly? Securin, which we're going to go into more detail, right? So securin holds chromosomes together, and if it's not degraded, will you have cell division? No. Okay. Right, so that would be bad. And so also S and M cyclins are degraded by APC. So that's another thing. You wouldn't be able to transition to your next phase of the cell cycle because you kind of get stuck. Yes? Securin involved in crossover, as in recombination? Well, okay. I'm seeing where you're going with this. So you're thinking because it keeps sister chromatids together and because recombination happens when they're paired, it must be. So my guess would be that if you didn't have securin, there'd be no opportunity for recombination. I think that's a very logical thought process. Yeah, I don't know any data, but I think that makes a lot of sense. Good job integrating multiple points of information. What about an APC that could not bind to CDC20? What would be the effect? Remember, CDC20 binds APC, and does that activate or inactivate? It activates it, right? So what would happen if APC could not bind to CDC20? Yes. Right. So you couldn't progress to anaphase, and well, basically you couldn't do anything that required degradation, right, of a substrate of APC. Would an individual would an individual with either of these mutations be viable? Would they ever be born? No, you would never even see someone with this probably, right? Because if you can't go through your cell cycle, how could you be alive? Right? Is cell cycle important for life? Thank you. Should I put that as an exam question? Yes. Question 1. Is cell cycle important for life? Okay. So would an individual with a mutated F-box be viable? F-box protein be viable? What is F-box protein? What does it do? It binds to phosphorylated substrates in the context of what? What does F-box protein associate with? A ubiquitin ligase called? Well, so CKI is a substrate that, when phosphorylated, is bound by an F-box protein, but F-box protein is part of a ubiquitin ligase complex. You could always remember that because SCF, the F stands for Xbox. I'm thinking of an exam question right now involving an Xbox. If you got an Xbox on sale on Black Friday. Okay, good. So would a person with a mutated F-box protein be viable? Why or why not? And these are such easy questions. I know you're just bored by my questions. You're bored. You're like, gosh, challenge us more, please. Yeah, maybe. No one?
No, I mean, you never break down your CDKs to progress to MS.
Right, so if you don't degrade your CDKs, will you be viable? And not CDKs, CKIs. CKIs. Because what do CKIs do? Oh, it's like dead people in here today.
They phosphorylate CDKs again.
CKIs, cyclin kinase inhibitors. They bind CDK cyclins.
I have a question then. But you know that there's other ways that So like even if that one like part like that one like regulatory mechanism is blocked, couldn't they still like rely on the other one?
I guess it depends on what. So the question was, and this is a great question. If one regulatory mechanism is blocked, couldn't there be other ways for survival, basically, right? And that's the importance of having multiple regulatory mechanisms. But that's where it depends on what regulatory mechanism is being blocked. So if you're not able to degrade CKIs, that's a tough one, right? Because that inhibits the whole complex. But let's say you weren't able to inhibit a phosphatase or a kinase, something like that. I could see there being redundant pathways. Maybe there would be other phosphatases or kinases that might be able to work in that place. But it's almost like if you have too much of one protein that binds and inhibits, it could be a worse situation. But it's I mean, my guess is, yes, there needs to be redundant pathways so that people can survive. Yep. Any questions about these practice problems? Okay. Okay, M phase. That's supposed to see. It looks so much nicer on my screen. That's just M phase. Here are mitotic spindles, your DNA. This is actually from some website where they a lab's website, and they focus on a protein that's involved in spindle assembly, and it's the yellow is some kind of protein that binds to the centrosome. Okay, so M phase. So we talked about S phase. Now what? We'll talk about M phase. So obviously, MCDK would be involved in initiating M phase, right? So I want you to remember this. I showed this to you before, where you have these gradual increases and then degradation, right? So here's your gradual increase starting at the end of S phase through G2 phase, right, of M cyclin, right? So you have to think about, remember we talked about this phosphorylation, inhibitory phosphorylation, all this stuff. This is why you need these mechanisms of regulation of these complexes, because you have this gradual increase of M cyclin, and it's going to just start associating with MCDK. And if that becomes activated before it's needed in like G2, that's not right? It needs to be, you need to wait until you get into M phase to activate. So what happens is you have phosphorylation by WEE1, which is this kinase that phosphorylates the active site of the CDK. And then early in mitosis, a phosphatase called CDC25 becomes activated. It removes that phosphate group, and that's what activates MCDK, right? So you've got your CDK, you've got your cyclin, and they're just going to bind to each other. But they're inactive until they're phosphorylated by CAK, which is the activating kinase. But then they're kept inactive by WEE1, right? So WEE1 phosphorylates active site, CAK phosphorylates an activating site, and it's not until MCDK, it's not until, I think it's a G2 MCDK, turns on, no, it's activated. Let's see. Why did I write possible activate? I just wrote that a few minutes ago. Well, so it's not until CDC25 is activated by phosphorylation that it removes this phosphate group and activates MCDK. So I wrote, so it's not known. So whenever I'm putting together a lecture, I actually get myself so bogged down with details. I do, because I'm always thinking, what questions are my genius students going to ask me? And so I was asking myself, well, how does CDC25 get activated? That's what I was asking, because if CDC25 removes the phosphate group early in mitosis to activate MCDK, so there's got to be some way to activate CDC25, and it's activated by phosphorylation. It must be by a CDK, and so I was trying to figure out which CDK is activating it, and so it said it's not really known, but it's possible that it's SCDK, which threw me off even more, because that makes no sense, right? Because SCDK, That's all. Well, that's active. Oh, yeah. See, there you go. That's active over here. It's active all the way through. So it makes sense why it would. Okay, so your MCDK is coming up, and your SCDK is still around. There you go. And that's how it gets turned on right there. But it was bothering me because you've got this accumulation, right? And you've got this SCDK that's active all the way through here. So why wouldn't it turn it on too early? I don't know. I guess it's not known, which is what the book said. It's not really known. But interesting things to think about. The whole point is that MCDK needs to be activated for mitosis to begin. Okay, so MCDK. So whenever I tell you a kinase is being activated, you need to ask yourself what's being phosphorylated, right? Who cares if a kinase is being activated if you don't know what's being phosphorylated? So things to think about, right? So what happens early in mitosis? Chromosomes condense, right? Does everyone remember that from every class you've ever taken in this building, probably? So chromosomes condense. Why do they condense? Why do they need to condense before they segregate? What's the point, other than that someone saw that under a microscope and told you that? What happens if your chromosomes do not condense before they segregate? I hear whispers.
They're not inherited by the daughter cells equally.
Okay, so they're not, if they don't condense, they're not.
Microtubules can't attach to them, so they're not condensed and they're not good.
So I hear they're not inherited equally because the mitotic spindle might not attach properly. That's like a not direct answer. That's not completely wrong, but not as direct as I want. Yes?
Could it just be like a sturdiness issue when they're being pulled apart?
A steadiness issue when they're being pulled apart. So they
Could
keep.
They like break when they're being pulled on or something?
They might break when they're being pulled apart. They might break when they're being pulled apart. No. So there's a comment about formation of the centromere. Centromeric DNA is actually what determines centromere formation. It's actually in the DNA structure itself. It is heterochromatin. So it is more condensed automatically, but not really directly. But the two comments over here were the ones that are most obvious, right? If you're remember what your DNA looks like in interphase before mitosis when chromosomes are not condensed, it's the whole nucleus is blue, right? So how would you pull those apart? How would you pull chromosomes apart if it's like a big who's ever made spaghetti but like you used angel hair and you forgot to put a little olive oil on it when it was done and you just threw it back in the pot and then you're trying to like serve it to people and the whole thing comes out at once?
I've definitely done that.
I've done that. I do it all the time. I like to use butter rather than olive oil. So my whole family has high cholesterol. But yeah, so think about it that way, right? If you don't condense your DNA, then it's going to be like trying to tear apart chunks of spaghetti that are all tangled up. It's totally like that. Do you want to tear apart your DNA when you're doing cell division? No, that's really bad. Broken DNA, is that good or bad? I would say broken DNA is really bad. It's like the fast track to death. So chromosome condensation is very important. And how does this happen? This protein called condensin is activated by phosphorylation by MCDK, and it is involved in condensing the chromosomes. So that's one target of MCDK, right? Okay, so what else does MCDK do? It activates mitotic spindle assembly early in mitosis. Remember this picture? Remember we talked all about mitotic spindle assembly, right? By the way, for those who are confused, on the exam, a lot of people seem to forget that microtubules do more than form a mitotic spindle. So that, I just want everyone to remember that that's a very important function of microtubules, but there's a whole lot that goes on in interphase. So forget mitosis, right? Interphase. That whole network, the train tracks that carry vesicles and all that, that's very important. So it's not just about mitotic spindle assembly. Anyways, I digress. Yeah, so MCDK activates mitotic spindle assembly. How do you think this is happening? What do you think MCDK is actually doing? So it needs to phosphorylate something. And you know you need microtubules and you need motor proteins. What do you think might be phosphorylated that's involved in mitotic spindle assembly? Take a guess. What did we learn about in terms of regulation of kinesins? Do you guys remember? At some point I said something, oh, activity is regulated by phosphorylation, and that's all I said. And you guys probably went, but here you go, right? Kinesins, very important. So we're going to go through that in just a minute. So it's dependent upon microtubule motor proteins, kinesin, and dynin. So you've got your plasma membrane, you've got your centrosomes and your microtubules, and then you've got a lot of motor proteins doing a lot of different things. So you've got dynins that are going to associate with other proteins in the cell cortex, as well as microtubules. Remember, they have cargo and they walk along the microtubules, and so they help to organize the microtubules and pull centrosomes apart. So you can imagine if it's attached to this cortex and walking to the minus end, it's going to kind of pull your mitotic spindle apart. You also have these interpolar microtubules that end up being cross-linked by kinesins, and they pull the poles together and push them apart, depending on which ones you're talking about. Then you've got these other kinesins, these chromokinesins. Chromo, bind to chromosomes. And they bind, their cargo are the sister chromatids. And they bind them and walk along, walk up to the plus end of the microtubules and help to push the chromosomes away. Remember, why would you want to push your chromosomes around? What's the point of that? What part of mitosis might that be involved in? Why would you want to push them around? Line them up. Metaphase? You ever wonder how they all get lined up so neatly? Or you guys just took it for granted? The miracles of cell division. It's all a miracle. Yeah, they get there somehow, right? So they are cargo, and the kinesins bind them and walk them to where they need to go. Then there are also other kinesins that bind. They have their motor domains on either end, right? And so they walk in the to the plus direction, and that ends up pulling centrosomes together. So all this pulling and pushing of your mitotic spindle eventually gets it so that the poles are in the appropriate location and that your chromosomes are in the appropriate location. And this is all regulated by MCDK and other mitotic kinases because they phosphorylate kinesin 5 and some of the other components. Before I go on, did everyone get that? Okay. You just almost can visualize all of these things pushing and pulling and pushing and pulling. The same way you have that happening to center the centrosome, you have that happening to push and pull your centrosomes or your mitotic spindle. So the poles are on either side. Okay. So centrosome duplication. Don't forget that needs to happen, right? So your centrosome is during interphase is just by by the nucleus, and your microtubules are radiating out, right? But by M phase, there needs to be two of them, and they need to be on opposite sides. And G1-SCDK actually initiates this process. And what happens is you get duplication. It's semi-conservative, actually, just like DNA replication. You get duplication as early as G1-S phase, but they don't move apart until M phase. Okay, so why do I have this here again? This is just to reiterate that the spindle assembly is dependent upon microtubule motor proteins, kinesin and dynin. And dynin is mostly promoting centrosome separation, which is increasing spindle length. And kinesin 14 and 5 pull the poles together. And these opposing forces are what result in the appropriate spindle length and position. So what do I mean about length, right? So there are these opposing forces, and they're generated by different motor proteins, and they actually play a role in the microtubule length in the mitotic spindle. So this was an experiment that they went over in the text. So you've got your normal, so your green, you've got your green microtubule. So this is just a microtubule. That's part of the mitotic spindle. And so normally it would be this length, and if you overexpress kinesin 14, you get a very short microtubule in your mitotic spindle. And if you overexpress kinesin 5, then you get a very long one. And so it's just that balance of the different motor proteins that results in the appropriate spindle length. Okay, microtubule stability. Did you guys think you were done with the cytoskeleton and you can just dump the information? See, this is what I mean. You can never forget anything. You have to integrate it all. Microtubule stability in mitosis. So do you think microtubules are more or less stable in mitosis? Remember, we learned all about dynamic instability. And now dynamic instability is regulated by different proteins, catastrophe factor, and these stabilizing MAP proteins. And they're going to be active at different levels, at different phases of the cell cycle. And so do you think they're more or less stable in mitosis? Yes. Do you think microtubules are more stable? Why?
I mean, because you want to maintain the mitotic spindles as much as possible, as opposed to in the normal cell where you have to shuttle back and forth to different parts of the cell. So in the case of mitosis, they're always in the form of mitotic spindles at the beginning, and then at the end, when they disintegrate and become the nucleus.
Okay, so you just said disintegrate. Were you talking about microtubule disassembly?
Yes.
Is that related to stability? Right? So you just actually answered you corrected your own statement, right? Because yeah, absolutely, disintegrate. Go ahead. Did you have?
I was just gonna say they would be less stable because they always have to change.
There would be less.
Makes it easier to change if they're less stable.
They'd be less stable because they always have to change. What do you mean?
Well, I would say,
Expand on that.
I would say not during the whole part of mitosis they are stable, but like there's different parts during the cycle where they need to be stable. But, like overall, they need to be less stable, so if they can break down the form as more stable. Does that make sense?
Maybe. So stability varies during the different phases of mitosis. I think you could say that's correct. So what part of mitosis do you think microtubules are the least stable? Cytokinesis. Go back a step. Cytokinesis is when the cells actually become two. But before that, they're extremely unstable. Anaphase, right? That's how chromosomes are separated is you have massive catastrophe. So we're going to go through that. So the question is, how is MCDK? So you have to think about, okay, well, there's all this catastrophe. Well, how do you think MCDK might regulate microtubule stability? So you have to ask yourself, what might MCDK be phosphorylating? And how is that regulating those proteins' activity? You guys better know this without me teaching it. Come on, integrate. Who got an A on the last exam? So you guys have no excuse. If you were a person with a 20, you have an excuse. But if you got an A, you better know this answer. Okay? What is involved with microtubule stability or catastrophe? What? Yep. GTP. Okay, GTP cap. What else? What proteins affect microtubule stability? You know, some of you guys are just quiet. That's okay. Huh? What did I hear? Staffman? Maybe. Wasn't what I was looking for, but why not? What else? Well, no. Something that's basically going to induce catastrophe is what I'm looking for. Catastrophe factors, right? They're these proteins that like to bind at the end of microtubules and tear them apart. No, that's a chop. No. Okay, so you have to think that MCDKs may be involved in inducing catastrophe, right? It's phosphorylating some kind of protein. Okay, let's talk about this. So the balance between stabilizing maps and catastrophe factors affect catastrophe frequency and microtubule length, right? That makes sense, right? If you've got lots of stabilizing maps, you're going to have long, stable microtubules. If you don't have those, but you've got lots of catastrophe factors, you're going to have like no microtubules, right? So here's an experiment where they looked at microtubules. Let's turn off the lights so you can see it better. So they looked at microtubules that were incubated with, they were fluorescent, it's not fluorescent there. So there's your centrosome and your microtubules, right? And they looked just normally during interphase, right? Nice, long microtubules. Remember, interphase, not many catastrophes, right? So this is how many catastrophes per minute. So less than half a catastrophe per minute. So fairly stable. No catastrophes for those who said there was no catastrophes. Fairly stable. In mitosis, so this is mitotic, you can see there's your spindle. Mitosis, a bit more catastrophe, right? So then they took this mitotic extract and they removed all the maps, and if by this I mean they're talking stabilizing maps, and a lot more catastrophes, which points to the role of these maps in stabilizing, right? You're gonna have more catastrophes. And then here they got rid of maps and catastrophe factors. And so you have very low level of catastrophes. So the point is, it's this balance of your maps, your stabilizing maps, and your catastrophe factors that results in increased catastrophe in mitosis. And we'll see what that has to do with. Well, before we get further with that, I wanted to bring RAN back up. Do you guys remember talking about RAN in the beginning of the semester? What was RAN associated with? Does anyone remember? Should I give a cumulative final exam? Huh? Anyone remember RAN? Who remembers RAN? Yes. Do you remember what it does?
I remember it, but I didn't want to say it out loud.
You don't want to say it out loud? Do you want to whisper it in my ear?
That's okay.
Yes.
Isn't it like nuclear import?
Nuclear import. Absolutely. Absolutely. You can think it's because it runs, it's a runner. It's like, the drug runners. I live in the city. I'm familiar with runners. You see these like really scrawny guys on bikes with backpacks early in the morning. I'm sure they're out here. They're just behind closed doors. They drive nice cars out here. Yeah, so RAN runs things in and out of the nucleus. There's a way to remember. Now, everyone will always know what RAN is because you'll remember crazy Dr. Ehrlich talking about a runner. So RAN not only is involved in nuclear import. Remember, it's a molecular switch. It is activated by chromatin-bound GEF. Remember that? Well, it's also involved in stimulating nucleation and stabilization of microtubules around chromosomes. Interesting. Would anyone have predicted that? Probably not. But it makes a lot of sense, doesn't it? Right? Why would you want to nucleate and stabilize microtubules near chromosomes? Why would you want to do that? We're talking about mitosis. Yes?
So they stay attached.
So they stay. Well, don't even talk about staying attached before they even attach, right? You want so that they can attach, they need to be in close proximity, right? You want to bring your microtubules that are going to be a part of your mitotic spindle in close proximity to your chromosomes. Does that make sense? Because how else are they going to ever find each other? So that's involved with, that's RAN. That is another role of RAN. So it also, not only does it run things in and out of the nucleus, but it runs microtubules to chromosomes. I crack myself up all the time up here. There's like a party going on up in here. So then motor proteins bind the chromosomes and align them, right? So you've got nucleation of your microtubules, and then you get this organization by these different motor proteins that then also bind. You get binding of your chromosomes by motor proteins that are attached to microtubules, and then this constant pushing and pulling and focusing of your poles of your mitotic spindle result in that alignment of your chromosomes in metaphase. That's how it happens. Do you guys know that? Do you guys love that? What you learned in 201 is starting to make sense? Is it starting to make sense? Or is it becoming ever and ever muddier? I remember watching the BBC after, what was that election where they had to do the recount and Gore should have won? 2000? 2000? And I remember I was listening to the BBC, and it was like the next morning, because we all went to sleep and we didn't know what was going on. The next morning, I just remember the guy on the BBC saying, like, things are about as clear as mud. And I was like, in the appropriate accent, of course. And it's true, and it stayed quite muddy for about 8 years, but that's a different story. I will not bring politics into my classroom. Okay, so we're talking about attachment of mitotic spindles to what? To the chromosome? Is that what needs to happen? Okay. So they're attached at the kinetochore, and so that's just the kinetochore is gonna bind right in the centromere region of the chromosome. It's like the waist of the chromosomes, right? Yes, no, someone. They don't all have waists. Anyways, so then they bind there, and that's how you end up having them separated. And so I kind of tried to explain this to some people earlier in the semester when we were talking about cytoskeleton and the mitotic spindle and how chromosomes are separated. And so this is the picture I was trying to draw for, I think it was in recitation, someone asked me. So the kinetochore is right over here, and it's attached to the chromosome right at the centromere, and it attaches to this ring which binds to the mitotic spindle. Because you must have been thinking to yourself, wait a minute, if microtubules are binding to chromosomes and it's catastrophe that's separating them, how do they stay attached to the chromosomes, right? Were you guys up late last night wondering that? Or were you just wondering when you were going to get your exam back? I promise, in 1 minute. So this is what allows the minus end of the microtubule to associate with the chromosome and catastrophe while still staying attached. Because this ring right here kind of slides back along, associated with microtubules. So, and that it still associates with the chromosomes and allows them to pull apart. We're going to stop right here so you have time to grab your exams.